Our title is Thrombosis Risk Assessment from COVID-19 Vaccine while large language model-driven tabular data extractions. This is also a collaborative study among Mayo Clinic and UMich. And in the past 4 years, the COVID-19 immunization have proved effective in reducing the severe illness and mortality. And we find at the national level, a rare instance of vaccine-associated thrombosis have been reported and studied in the past several years. A thematic globally scoped comprehensive analysis have yet to be conducted. So this study leveraged the large language model to investigate 3 COVID-19 vaccines induced thrombosis risk across different countries using evidence extracted from tables of public available literature. And this is our pipeline or method. Totally there are 3 steps. The first 3 parts. The first part is the article corpus collaboration, following the our method of table extraction with large language model. And the third part is thrombosis recognition with LLM. And this is the first part. We collect the data and from the PubMed. So we focus on the widely administered COVID-19 vaccine, Pfizer, Moderna, and Janssen. And we collect the 10 data from PubMed. And with we make query with the COVID-19, COVID-19, and coronavirus, and we get over 450,000 articles. And then filtered with the keywords of Moderna, Pfizer, and Janssen. We collect over 8,000 articles. And the last step is we will continue to screen the articles with either event and thrombosis. And we obtain 37 papers. All those papers are PDF formatted. The next step is the table extractions. I think that if we extract the table from PDF file directly, there is 4 challenges, I think. And the first challenge is the table tag detection. It is very hard to detect table from the PDF file. The second is the table issue. And for example, for the right top example, for in the table consists of 4 small tables. For the layout, the page layout issue, they also divide into several small ones. It looks like one table. Actually, there is 3 or 4 tables in it. And the third challenge is the split page tables, because some table may be very large. It will span multiple pages. And you could see from the right bottom example, this is one table, and there is they use 2 pages. You could say to continue the next pages, and then on the next pages, you could say for the table 1 continued. And the last challenge is the large language model issue. So for the PDF file, it is not a text format. So for the large language model, if we use it, we want to use it. The first step with the tool notation. The tool notation is based on the text. So the first step is how to extract the text from the PDF file. If we fetch the binary string to the large language model, yeah, it's a very great, it's a very good challenge to for the large language model to extract anything from the PDF documentation. So we designed these pipelines. Our method is we did not directly extract the table from the PDF file. We want to we want to convert to the text format file first. The one important text file is HTML. And if we directly convert the PDF documentation to the HTML file, and the tags will be lost. And so we adopt two steps to convert to convert the PDF to HTML. The first step is to convert the PDF to a Word file, Microsoft Word. We use the PDF to DOCX library, and this purpose is to preserve the table and the tags within the PDF file. The second step is to transfer the PDF, the Microsoft Word into HTML with the pypandoc libraries. And this step, its purpose is to generate the HTML table and tags. And the third step is to extract the table from HTML. And we will design some prompt and use the zero shot and feed to the Q1 large model. The last step is to mine the thrombosis information from tables and the whole paper, because for some not all the useful information about thrombosis we could find in the table. For example, the total number of the population and the doses, and some table did not list them in it. So we need to mine this information from the whole paper. We also use the zero shot. And in our experiment, so we prepared two data corpus. The first is 20 papers, and we use it to test our method. And those papers covered 4 types of tables: the single line headers, multi-level headers, hierarchical headers, and vertical headers. And the second corpus is we use it to test our it's the 37 thrombosis articles. And we compared our result with some SOTA methods. We compare with the tabular-extraction and Kimlot and the PDF miners. Compare with the tabular and Kimlot, there is a great improvement for our method. And for the compare with the PDF miner, our result is also competitive, and especially for the record number and the captain. Yeah. And for this table, we use the extract information to do some statistical analysis over the thrombosis related to 3 COVID-19 vaccines: the Pfizer, Moderna, and Janssen. And we could see from this table, we could see there is 2 different disclosure units. One is the population, the second is dose. And for the content, all those paper, all those data came from the North America and Europe. And from the age, it is very hard to say. Some paper mention it, some paper did not mention, or it is confused. So I did not list them. And for the thrombosis, we could say for the Moderna and the Pfizer, its risk is relatively low compared with the Janssen. The Janssen is over 3 times than the Moderna and Pfizer. And we could get a similar result from both the dose and the populations. And for the Europe, we did not find any use for the Janssen. And as a conclusion, and we created a thrombosis corpus from the public available literatures. We developed a robust PDF to HTML large model-based pipeline that constructs to extract the complex tables. The third is the reported thrombosis risk associated with the Janssen is more than 3-fold higher than that of being Pfizer and Moderna. This is some references. Okay, that's all. Thank you.
Great. Well, thank you very much for the excellent talk. Do we have any question for George? Just maybe one quick question. If not, maybe I can ask. You use the PDF as the input. I wonder why. Well, you could go directly from HTML. So most of the articles these days have their XML version available, and you don't have to extract those tables using a special tool. Have you tried that way?
I tried. I compared. Yeah, from the PubMed. I can also download the HTML directly, and it result. It is a little better than our result. Because for some of the HTML, maybe they generate manually. The For our method, we produce the HTML automatically. This is the major
Okay.
difference.
But I don't know how many of those 37 articles you use are also indexed in PubMed Central. And if any of them are in PubMed Central, they have XML version, then you can clearly cut out those table sections. You can take a look.
You mean for the 33 articles?
Yeah.
And yeah, this article is, it is not too much, for the data set. I think it is caused by the adverse event. For the third screen keywords, if we remove the adverse event, maybe we can get more articles. So that is, this is the preliminary result. So I did not, maybe this is for the next step, I will, remove the adverse event from the keyword list.
Yeah, I think we can follow up on that issue.